Hi friends, welcome to our channel Way to Success. இந்த வீடியோவில் PGTRB Zoology Unit 1 Branchiostoma short notes பார்க்கலாம். Branchiostoma, Ambiaxis, Classification, Phylum Chordata, Subphylum Cephalochordata, Class Leptochordae, Family Branchiostomidae. Introduction. Ambiaxis is a protochordate. It is popularly known as lancelet. It is discovered and described by a German taxonomist, Pallas. He considered it as a mollusca and named it Limax lanceolatum. Later, Casta, 1834, named it as Branchiostoma. The name Ambiaxis proposed by Yarrell in 1836. This animal is peculiar in that the anterior end of the notochord extends into the head. Hence, it is included in the subphylum Cephalochordata. Its scientific name is Branchiostoma lanceolatum. It is marine animal, burrowing. It lives in shallow waters. In the daytime, it remains buried in sand in an upright condition. At night, they leave the sand and leads a free swimming life. Structure of Ambiaxis. It is spindle in shape and laterally compressed. Both the ends are pointed and looks like a lancet. It is 4 to 6 cm in long. The body is divisible into anterior trunk and a posterior tail. It has no head. The anterior end has a pointed process called rostrum. A frill-like membranous enclosure is present beneath the rostrum is called oral hood. The oral hood opens to the outside at the anterior end by an opening called mouth. The cavity enclosed by the oral hood is called vestibule or buccal cavity. The margin of the oral hood bears 20 or more tentacles called oral cirri. The ventral surface of the anterior two-thirds of the body is flat and called epipleur. The lateral margins of the epipleur are produced into two thin folds called metapleural folds. Ambiaxis has three external openings. They are the mouth, the atriopore, and the anus. Ambiaxis has three fins. They are dorsal fin, that is mid-dorsal line. Caudal fin surrounds the tail. Ventral fin, ventrally, caudal fin extends forward about 1/3 of the length of the body. The dorsal and ventral fins are supported by a skeletal framework called fin-ray boxes. Fins are used for locomotion. Body wall of Ambiaxis. The body wall is formed of many layers. They are: first layer is cuticle layer, thin outer layer. Second one, epidermis. It is made up of single layer of cells. Epidermal cells or columnar cells are present here. Third, basement membrane. The fourth layer is dermis layer. It is formed of connective tissue fibers. The cuticle, the epidermis and the dermis together constitute the skin. The muscles are located beneath the skin. They are metamerically segmented. The muscles are in the form of E-shaped blocks called myotomes. They are about 62 myotomes on each side. The muscles are internally lined with peritoneum. Atrium. The spacious cavity lying inside the body is called atrium. It surrounds the pharynx, esophagus, and intestine. It is lined with ectoderm. It opens to the outside by atriopore. The gill slits of pharynx open into the atrium. The ventral wall of atrium is the epipleur. The main function of atrium is to give protection to the gills. Next we can see about coelom. Coelom is the body cavity lying between the body wall and the alimentary canal. It is lined with the coelomic epithelium. In the pharyngeal region, it is reduced to three types of coelomic spaces. The three types of coelomic spaces are: first one, a midventral subendostylar coelom running below the endostyle. Second one, two dorsal longitudinal canals lying above the pharynx. Third one, the vertical coelomic canals in the primary gill bars. Next we can see about notochord. Notochord is the back chord. It is a gelatinous cylindrical skeletal rod containing vacuolated notochordal cells located on the back. It is the axial skeleton. The notochord is situated above the alimentary canal and below the nerve cord. It is covered by notochordal sheath. The notochord is made up of fibrous connective tissue. Inner to the notochordal sheath, there is a thin elastic membrane called elastic interna. Below the elastic interna, there are two canals, dorsal and ventral canal. The notochordal cells are arranged in a longitudinal row. In ambiaxis and vertebrates, the notochord develops from mesoderm. But in balanoclastes and ascidians, it develops from endoderm. The rostrum supported by the notochord helps in burrowing. Digestive system of ambiaxis. The digestive system includes the alimentary canal and the digestive glands. The mouth is a wide opening located below the rostrum. Oral hood bears about 20 finger-like process called oral cirri. The oral cirri function as a sort of seed during feeding. The oral hood encloses a spacious cavity called buccal cavity. It is lined with ectoderm. The ectodermal lining is folded to form a number of thick finger-like ciliated tracts. These tracts together called wheel organ. It is used for creating whirling water currents. Mid-dorsal tract of wheel organ is larger and is called Hadsex groove, which ends in a glandular depression called Hadsex pit. The posterior end of the buccal cavity has a vertical partition called velum. The wall of pharynx perforated by a row of 180 vertical slit-like opening called gill slits. The wall of pharynx lying between the gill slits called gill bars, that is the primary and secondary gill bars. The primary and secondary gill bars are interconnected by a numerous horizontal partitions called synapticula. The floor of pharynx has a shallow longitudinal groove called entostyle. Entostyle has two functions. It secretes mucus and creates water current. The pharynx leads into a short tubular esophagus. The esophagus leads into the intestine. The intestine is divisible into an anterior wide midgut and a posterior narrow hindgut. It opens to the outside by anus. Digestive gland. Ambiaxis has a single digestive gland called midgut diverticulum or hepatic diverticulum. Digestive secretions are enzymes, that is lipase, amylase, protease. Feeding habit. Ambiaxis is a carnivorous animal. It feeds on microscopic planktons and protozoans. It feeds in three ways: ciliary feeding, filter feeding and mucus feeding. Next, process of digestion. The water current carries food materials. The water current enters the pharynx via mouth, buccal cavity and enterostomy. The mucus gland cells of endostyle secretes mucus. The mucus moves upwards along the lateral walls of the pharynx. The mucus cord is then passed into the esophagus and then it passes into the midgut. From the midgut, it enters the midgut diverticulum, then it enters the hindgut. Digestion occurs inside the midgut and the hepatic diverticulum. Digested food is absorbed in the hindgut. The undigested material is eliminated through the anus. In ambiaxis, digestion is both extracellular, it occurs in the midgut, and intracellular digestion, it occurs in the hepatic diverticulum. Next, respiratory system of ambiaxis. Ambiaxis has no well-defined respiratory system. Respiration occurs by diffusion through the body surface. The gill slits of ambiaxis play a minor role in respiration. Circulatory system of ambiaxis. Heart is absent. Blood is colorless. No corpuscles and pigments. Blood help in transportation of food and excretory products only rather than gaseous exchange. There is no structural differences between the arteries and veins. The chief blood vessels and its branches are as follows. First one, sinus venosus. Below the posterior end of pharynx, a thin-walled sac-like structure is present called sinus venosus. It collected blood from different parts of the body through veins and pumped forward into the ventral aorta. Second one is ventral aorta. It is a large median longitudinal artery, arises from the sinus venosus and runs midventrally in the wall of the pharynx below the interstitium. A series of paired lateral afferent branchial arteries arises from ventral aorta, running through each primary gillbar of pharynx. At the base of gillbar, each artery forms a tiny bulb called bulbillus, which helps in circulating blood. The branchial vessels of primary gillbars connected to secondary gillbars by transverse vessels. The afferent branchial vessels leave pharynx dorsally as efferent branchial vessels, which open into a lateral dorsal aorta. Before opening, each efferent vessel forms a small capillary network called nephric glomerular sinus and nephridium. Third one, dorsal aorta. Two longitudinal vessels run, one on either dorsolateral side of the pharynx, called right and left lateral dorsal aortae. They continue anteriorly as internal carotid arteries to supply blood in oral hood. Both lateral aortae unite just behind the pharynx and form a median dorsal aorta. Median dorsal aorta run posteriorly between the notochord and the intestine and enters the tail region called caudal artery. From the median dorsal aorta, many intestinal arteries arises which supplies blood to intestine. In dorsal aortae, blood flow backwardly. Next, the fourth vessel, subintestinal vein. It is formed by the midventral caudal vein and the intestinal vein. Caudal vein collected blood from tail region, while the intestinal vein from the intestine. Blood flows in it anteriorly. Next one, hepatic portal system. The subintestinal vein continues anteriorly as the hepatic portal vein, which run ventrally along midgut diverticulum and form capillary network. Hepatic vein collected blood from midgut diverticulum, run dorsally and bend downwards to join the sinus venosus. Sixth one, cardinal veins. Blood from ventrolateral regions of the body is collected on either side by anterior and posterior caudal vein and sent to sinus venosus. The seventh one, parietal vein. Blood from dorsal body wall is collected by a pair of parietal veins. They run above the intestine and trunk, ventrally join the sinus venosus. Circulation of blood. The blood flow anteriorly inside parietal, subintestinal, posterior cardinal veins and ventral aorta, where it flow posteriorly by lateral and median dorsal aortae and anterior cardinal vein. Next, excretory system of ambiaxis. The excretory system is formed of three types of organs, namely, first one, segmental protonephridium. There are 90 pairs of nephridia. Each nephridium has 500 solenocytes. Each solenocyte is a round cell with a nucleus and a long tubule. It is developed from ectoderm. It is similar to flame cell of Annelida. Second one, Hatz-Sachs nephridium. It is a large nephridium situated in the roof of the buccal cavity. It bears numerous solenocytes. Third one, brown funnels. Brown funnels are excretory organs of ambiaxis. They are brown in color and funnel shaped. There are two brown funnels. Mechanism of excretion. Solenocytes extract nitrogenous waste from blood and pour into atrium through nephridiophore. From the atrium, it escapes through atriophore. Nervous system of ambiaxis. Ambiaxis has a hollow neural tube. It extends throughout the length of the animal. The canal situated inside the neural tube is called a neurosil. The neural tube gives out paired spinal nerves which are arranged segmentally. Next we can see about sensory organs of ambiaxis. First one, eyes. Ambiaxis has numerous eyes along the entire length of the neural tube. Second one, pigment spot. The anterior wall of the cerebral vesicle has a pigment spot. It is a thermoreceptor. Third one, infundibular organ. It is a depression in the floor of the cerebral vesicle. Fourth one, holocuspid. It is a ciliated depression located on the dorsal side of the cerebral vesicle. It acts as a chemoreceptor. Next, reproductive system of ambiaxis. The sexes are separate, but there is no sexual dimorphism. Gonads are segmentally arranged. 27 pairs of testes in the male and 27 pairs of ovaries in the female, arranged one in each segment from 25 to 51. Once gametes are mature, the gonads rupture and release gametes to atrium and escape into seawater, where fertilization occurs. So here external fertilization takes place. The fertilized egg undergo cleavage and gastrulation and finally develops into an ambiaxis. Affinities of ambiaxis. First one, affinities with Annelida, bilateral symmetrical, metamerical segmentation, protonephridia, coelom, closed circulatory system and filter feeding. Next, affinities with mollusca, ciliary mode of feeding. Third one, affinities with echinodermata, asymmetrical body, enterocoelous coelom. Affinities with chordates, dorsal tubular nerve cord, gill slits, notochord. Affinities with hemichordata, gill slits and gill bars, filter feeding, enterocoelous coelom, numerous gonads. Next one, affinities with urochordata, ciliary mode of feeding, atrium with atriopore, pharynx with gill slits, endostyle, dorsal lamina and peripharyngeal bands. Respiratory mechanism. Affinities with cyclostoma, fish-like body, continuous dorsal median fin, gill slits, and endostyle. Next one, affinities with other vertebrates, true coelom, midgut diverticulum comparable to liver, hepatic portal system, spinal nerves with two roots. Next, the special characters of Ambryoxus are large pharynx, numerous gill slits, atrium, notochord extending far into the head, oral hood, asymmetry. Thank you for watching this video.